Hello, my name is Arun Bhunia. I'm a professor of food microbiology at Purdue University, and I'd like to thank the organizers for inviting me to discuss about some of those biosensor technologies we are developing at Purdue University for detection of foodborne pathogens. Before I talk about biosensor technologies, I'd like to give you a little background about foodborne pathogens and the disease they cause. And most of the time, the foodborne diseases are characterized by vomiting, diarrhea, or dysentery or gastrointestinal complications. And those are caused by bacterial as well as viral and parasitic pathogens. But in addition to this acute disease, bacterial pathogens also cause, or microbial pathogens also cause numerous chronic diseases like arthritis. They also could cause kidney disease like by Shiga-toxin E. coli, also can cause hepatitis or jaundice, Guillain-Barré syndrome. This is one of the disease. It's a debilitating disease due to Campylobacter infection. Also can have central nervous system disorder or meningitis or encephalitis caused by Listeria or BSc or bovine spongiform encephalopathy, Nipah virus. Miscarriage or stillbirth or neonatal infection can be caused by Listeria monocytogenes or Toxoplasma gondii. Paralysis due to neurotoxin produced by Clostridium botulinum, and sometimes also toxins of seafood origin. In addition to that, cancers and malignancies can be caused by foodborne toxins like mycotoxins, and allergic responses could be caused by seafood toxins. So foodborne pathogens causes varieties of disease. Therefore, we need to develop methods that will help us to detect each of those organisms. If you look at the significance of these foodborne pathogens, especially in the US, we have about 48 million illnesses and about 3,000 deaths annually due to foodborne infections. And of these 48 million illnesses, known pathogens, they represent about 24 organisms responsible for 9.4 million illnesses and about 1,300 deaths. And the most striking is that unknown agents responsible for about 38.4 million illnesses and 1,600 deaths. And so our goal in this case would be that we have to do something in order to detect these unknown agents, because most of the time our focus is on known foodborne pathogens. And if you look at those unknown agents actually that represent various traditional microorganisms like Aeromonas, Plysomonas, or Mycobacterium, or there are also some known agents like Clostridium difficile, Trypanosoma, and also there are some emerging pathogens. For example, we had O104H4 outbreak in Germany through sprout, and these are some of those emerging pathogens that can also cause problem. So if you look at overall, so we need to develop technology platforms that would help us to not only the known pathogens, we need to also develop technology platforms for detection of unknown pathogens. Then we have a better control of foodborne pathogens and the related diseases. The cost is also enormous. It costs about $78 billion and $45 billion for unknown agents. And the cost normally associated with lost work, death, medical bills, product recalls, bankruptcy, lawsuits, and so on. If you look at globally, there are about 1.8 million deaths due to food and waterborne diseases. So if you look at the overall farm to fork food safety and the food supply chain, most of our products start with the farm, and then products move to processing plant, and this is where most of our testing is done. And then the product is shipped to for storage, then transported through trucks or other means, and then distributed to the retail distribution or restaurants, and then ultimately to the consumers. So if the detection is done in the processed product, so if pathogen contamination or their growth happens during this supply chain, probably we have no way of monitoring or controlling them. On top of it, also the products directly transported from farmers market to the consumers, and in those cases this farm products, meat or produce, fruits and vegetables, they may also carry pathogens directly to the consumers. So overall, we need to develop technology platforms that would help us to monitor pathogen presence in every aspect of this farm to fork food safety supply chain. And if you look at the detection methods, and one of the concern we have is that most of the products are negative for target pathogens, and both ready to eat as well as raw products. So we develop technology platforms that help us to monitor the absence of pathogens. So for detection approach, there is not a single approach that can allow you to detect various foodborne pathogens. So we have categorized this pathogen testing approach based on the time and the specificity as well as sensitivity. So overall, culture-based or traditional conventional methods requires about anywhere 3 days to 14 days, and it is a time-consuming method, but it is highly specific as well as highly sensitive. And so therefore people are looking for rapid methods that can be completed within 24 to 48 hours. These methods are somewhat specific and also highly sensitive. And there are methods ultra rapid, which can be completed within 8 hours or less. They are also reasonably specific and sensitive. And most importantly, people are also looking for real-time methods that could be used online or inline, and that would yield result within a seconds or minute. But one of the drawbacks of these methods are they are less specific. However, they might be sensitive. And in recent years, we are also looking for technology platforms that will help us to for rapid high throughput screening. And if we could achieve that within seconds to minutes, that will be the desirable as well as highly specific. Nevertheless, I'll talk about those different biosensor technologies that would be able to complete it within reasonable time frame that may be associated with culture-based methods as well as real-time online methods. So in reality, we are looking for biosensor-based approach in two-step method. And in the first step, we need to develop high throughput screening, and that would yield results very quickly, preferably real time or near real time. And it should be high throughput, should be able to screen the product for multi-pathogen presence or absence, and it should be inexpensive so that product could be released immediately. If we find any sample likely to contain any pathogens or toxin, those products in second step could be tested rigorously for presence of a specific type of pathogens. So if you take two-step approach, that would yield pathogen testing much quicker and faster and in an inexpensive way, and that would be much reasonable approach for overall food safety. And so in a recent days, with our colleagues, we edited a book called High Throughput Screening for Food Safety Assessment, and this would be published sometimes in later this year, and this would be a good resource for people who are interested in finding high throughput screening technologies for foodborne pathogens. So what are those technologies? And high throughput screening technologies, for example, DNA microarray, and that can help detect multiple different organisms, protein or antibody microarrays, also multiplex PCR that targets multiple pathogens, and multi-top based mass spectrometry methods also becoming popular for detecting presence or absence of microorganisms or pathogens, and whole genome sequencing is becoming more inexpensive, and this helps us to identify the organism, and that could be also used for tracking of pathogens in the food supply chain. And lastly, the biosensors, and I'm going to discuss a little bit more detail about some of these biosensors that include level-free sensors as well as level-based sensors. Level-free sensors, generally these are spectral-based methods. It doesn't require any labeling reagents or probes, And so it depends on, however, a spectral library. For example, hyperspectral imaging system, or Fourier transform infrared spectroscopy, or Raman, or light scattering technology. This is one of the technology we are developing in our laboratory. I'm going to discuss a little bit more in detail. And label-based sensors that include fiber optic sensors, SPR, QCM, or impedance. And these methods require some DNA probe or antibody probe, as well as some labeling reagent that would provide specificity for their target organism or toxins. So ideally, when you want to apply biosensors, we want to apply directly to the sample, and that way we can get results immediately. But we haven't arrived to that situations where we would be able to, like in a grocery store, when you check out your produce or a product for price, and we like to have a sensor that would help you to find out the presence of any pathogens, ideally like E. coli or Salmonella. So that will be ideal scenario to use biosensors for quick screening of product before consumers obtain that product. But we haven't reached there yet. This should be something we have to look for in future research. And so in USDA ARS laboratory in Beltsville, Maryland, they are developing technology for online inspection. In that case, they have this sensor sitting on the top of a conveyor belt. As produce, in this case apple, moves through the conveyor system, it in real time can monitor presence of fecal contamination or any other contamination, and then those products could be immediately removed from the line. And having a indication of fecal contamination implies there might be pathogens present in those samples. Similarly, for poultry carcass screening, and in the conveyor belt, you can see this real-time acquisition of image of the whole bird, and they are looking for any pathological damage or fecal contamination on the surface, and then they can be immediately removed from the line, and that will reduce chance for contamination with microorganisms of harmful in nature. And so this type of online inspection is very powerful and can do very quick screening, and that will help to ensure food safety in our products. However, biosensor technology is still primarily or largely concentrated in the laboratory setup. So in order to use biosensors, we have to separate, concentrate, or enrich microorganisms, test organisms from sample. And there are a number of methods are used, like bioseparation methods, filtration, or immunomagnetic bead-based methods, or affinity methods, or dielectrophoresis. These allows separation of microorganisms or test organisms from sample, and then we apply biosensors. And I'm going to give you examples of some of these biosensors our laboratory is involved in developing at Purdue University. For example, in this case, Professor Applegate in the Department of Food Science, he has engineered bacteriophage that actually produces light when it infects bacteria. So in this case, in the left panel, you see the ground beef slurry, and it has been inoculated with E. coli 15787, and this bacteriophage is specific for infection of E. coli 157. And as you can see in this video, as the infection of bacteriophage take place to the bacteria, and then it's going to produce light. And this is a time-lapse video over 16 hours, and you can see the bacteriophage infection of E. coli 15787 cells produce light, and that can be easily captured by a camera. And so this type of online rapid screening of the food sample on the bag would be extremely useful for microbiologists to quickly decide if the product is safe or needs to be discarded. So we are also developing on one sensor called fiber optic sensor, and in this case, this sensor depends on the availability or the capture of microorganisms or target pathogens using antibody or receptor molecules on this optical waveguide. You can see it's about 4 centimeter long, and once it captures the target pathogen, there is a second antibody or second molecule that is conjugated to fluorophore molecules, and once this sandwich is formed, and this can be easily detected when the laser is passed through this optical waveguide. And in this case, when the laser hits this waveguide and this excites this molecule, and part of this evanescent wave returns back, and in real time you could monitor this presence of pathogens on the sample. This is a portable fiber optic device called Receptor. It's built with microfluidic setup. So this cartridge fibers in a cartridge goes in this slot, and then the liquid is moved through test sample, and then you can monitor in real time presence of any target pathogens as you see signal generated from this device. We applied this sensor for detection of Listeria monocytogenes. In this case, we used antibody for capture of Listeria monocytogenes, and then we have aptamer that's conjugated to fluorophore, and this will bind and form a sandwich. Aptamer is a short stretch of DNA that's highly specific for target protein of interest or molecule on the bacterial cells that can be developed and screened. And using this sandwich configuration, we are able to detect Listeria monocytogenes even in presence of E. coli or Salmonella. Also, this fiber optic sensor did not show much cross reaction with other non-pathogenic Listeria or other organisms. We also use these fiber optic sensors using immunomagnetic bead separation method. Immunomagnetic bead separation allowed capture of Listeria monocytogenes from food sample. Once they are captured, then we detected those microorganisms using fiber optic sensor. In this slide, you see this magnetic bead is captured one bacterial cell, and once we collect these organisms, this concentrated this Listeria monocytogenes cells from food matrix, we can verify that by fiber optic sensors. As you can see, this fiber optic sensors is highly specific for Listeria monocytogenes as well as Listeria ivanovii. This organism is also pathogenic to humans as well as animals, and this sensor again highly specific and did not show cross reaction with other microorganisms including Enterobacter or Salmonella or Staphylococcus or E. coli. We monitored the detection limit for these biosensors, and we've observed that we can detect about 100 Listeria monocytogenes cells if present in the food. So this sensor, fiber optic sensor, is highly sensitive and can be used with food sample for detection of low level of organisms. This slide actually summarizes some of those fiber optic sensor based methods for detection of Listeria, E. coli and Salmonella. And Listeria monocytogenes, as I mentioned, the detection limit is about 100 to 1000 cells, and we can detect less than 24 hours, and we have tested with hot dog, bologna and soft cheese. E. coli, we could detect about 1000 E. coli 15787 cells and from ground beef, and we can do it under 4 to 6 hours of time. And same way, Salmonella Enteritidis also were detected around 10,000 colony forming unit from chicken or egg, and this is also can be completed under 8 hours. So these methods clearly suggest that this fiber optic sensor can be used for rapid detection of foodborne pathogens from various food matrices. We also combine these 3 fiber optic sensors for detection of all 3 organisms if they are present in the same sample. The approach would be the food sample has to be enriched in a universal enrichment broth. This is called Salmonella E. coli Listeria enrichment broth we developed in our laboratory. And after enrichment, the sample is briefly processed and was delivered directly to the fibers for detection of these organisms. And as you can see, this method can detect Listeria monocytogenes in various meat products that were inoculated with Listeria monocytogenes, E. coli or Salmonella, and then we obtain specific reaction. So this study clearly shows that we are able to detect multiple pathogens if they are present in the same sample with a single enrichment step. Next, we looked at cell-based biosensor. This is also called functional biosensor, and essentially this sensor monitors real-time interaction of pathogens with the living tissues or mammalian system. Historically, if you look at it, the miners used to take canary bird into the mine for monitoring carbon monoxide, and these birds are highly sensitive to carbon monoxide. If this bird die or show any kind of illness, that indicates that the mine contains carbon monoxide. And so this is an example of functional biosensing. In modern days, we are looking at if you have a test organism like bacterial cells or pathogens or toxins, we will expose that to animals or humans, and we look for mortality and morbidity. Obviously, for ethical reasons, we continue to look for alternative method, and in that case, we are looking at cells derived from these animals or humans and expose these cells to microorganisms and then look for mortality or cell damage. In this case, you are looking at kidney cells, healthy kidney cells. When they're exposed to certain toxins, you can see the cells change their morphological appearance and the cells are dying. And in this case also, these lymphocyte cells are severely damaged due to Listeria monocytogenes infection. And our goal is to develop biosensor to monitor changes in these healthy cells due to interaction of pathogens or toxins. So you can essentially, one can engineer a mammalian cell with the specific receptors on the surface of the cells, on the membrane, that can target specific agents. In this case, you could target toxins like membrane active toxins or diarrheagenic toxins, or superantigens or neurotoxins like Clostridium botulinum toxins. If the cells respond to any of those specific molecules due to specific receptor, that the changes would be monitored in the form of color or fluorescence or luminescence or impedance. So the detector can monitor the healthy cells to the damaged or injured cells in the form of different indicators. So what we developed actually called cell-based sensor using three-dimensional cell configuration. In this 96-well plate, we grew our cells, and the cells are immobilized within the collagen matrix, and the cells has the opportunity to grow in all three dimensions. And so once the sample is exposed, the cells are exposed to different types of samples, and then you can monitor the color change using a spectrophotometer or spectrophotometer. And even one can also look at the color change in the wells. And for example, listrolysine produced by Listeria monocytogenes is produced yellow color. Same way, cytolysine, Bacillus toxin, or alpha hemolysin from Staph aureus produce positive color, or phospholipase C from Clostridium perfringens produced yellow color. Whereas this specific toxin, this specific cell types are not sensitive to cholera toxin, so that yielded negative result. So you could literally read the intensity of color change using a spectrophotometer, or you could look through the wells for color change. And that gives you a very quick screening tool for multiple test organisms or pathogens in one plate. Next, I'm going to talk to you about light scattering sensor. This is also called label-free spectral-based biosensor. One, in a microbiology laboratory, traditionally we look for bacterial colony on the plate for their color or appearance, sometimes even also smell. And that can give us some idea to microbiologists about the potential presence of certain organism. And what we developed is called laser light scattering, and this is called BARDOT. That stands for Bacterial Rapid Detection using Optical Scattering Technology. So essentially in this method, what we have is the laser, and once the laser is shined on a petri dish with the bacterial colony, and that produces a beautiful scatter signature. And these scatter signatures are extremely unique for different organisms, and then one can take these images using digital camera. And as you can see in this figure, here the students are working, shining laser on this petri dish, and you can see the scatter signature on this whiteboard. And initially we looked at Listeria monocytogenes, since we had a huge collection of Listeria. If you look at Listeria monocytogenes patterns, that's unique, different from Listeria ivanovii, Listeria inaqua, Listeria siligri, walsameri, and greyi. So these data clearly show is that even visually one can differentiate scatter signatures of different Listeria species and can detect or identify what colony or what Listeria present in that single plate. And over the years we established this technology, and in early 2003 and 2004, when this is our initial platform on the breadboard, and then once we put the laser on the top, and then the petri dish in the middle, and then the CCD camera down below to acquire images directly to the computer. Then this system was placed in a box, and then the system beginning to get sophisticated and smaller, and then we started building an incubator with the system, so that one can load the petri dish in the incubator, and once these plates are ready, the system can automatically collect those plates and read it, and so the user do not need to be in that location to run the system. So this is an automated system with a built-in incubator. So in the present day, what we are doing, actually we have built a benchtop model, and the company who has marketed, commercialized this unit called Advanced Bioimaging Systems, and this has a 2-plate reader. It can automatically read 2 plates simultaneously. This is a system built with 770 plate incubator, and again this is an automated device with 700 large number of plates, and the system can be programmed to collect these plates and scan for presence of any potential pathogens. And these are some of the more advanced the instrument. As you can see in this one, the 770 plate holding incubator system with 4 readers, and it's quite a large device, large system. And this one is the 44 plate holder. The incubator is in the bottom of the device, and so once the plates are ready, the system can collect those plates and read it, and can give you identification of specific microorganisms present. Here is a demonstration of this system, how it works. This quick video will show you that in real time this system is going to first take the image of the plate and locate each colony on the petri dish. As you can see, first it scan the plate, and once it finishes scanning, it will get the map of the plate, locating each of those colonies, and it has a parameter setup. This is to detect only 1 millimeter diameter colony, and once it has those parameters fixed, it can move from one colony to other, and it will collect this scatter image in real time. Once it collects those scatter images or signatures, it will collect in the database, and then we can compare this image with the images stored in the database, and based on that it can give you identification of the organism present in the plate. So it can be developed for specific pathogens, or some of the organisms we are working in the laboratory focused on Listeria, Salmonella, E. coli, Vibrio, Bacillus, Staphylococcus, Campylobacter, and so on. So here are some of those images, scatter images of Listeria as they grow on BHI agar plate, and you can see the different Listeria species producing different type of scatter patterns over different time period. We also tested with the Listeria when they grow on the MOX, modified Oxford agar plate, and also can be used to detect Listeria. In this experiment, you will see the Listeria was present in the hot dog, and we collected those Listeria cells using magnetic bead, and those colonies are allowed to grow on the plate. And after scanning, you can find out some of them are Listeria inaqua, which is non-pathogenic. Some of them are Listeria monocytogenes, producing two distinct colonies scatter signatures. So this study shows that clearly one can use light scattering device to find out the presence or absence of any pathogenic organisms on the plate. We also tested this system with Salmonella, and this is Salmonella when they grow on XLD agar plate, and we actually initially tested this Salmonella on different types of selective agar plate, and this was done on brilliant green, XLD and XLT4, and as you can see, depending on the media, they will produce different type of scatter signature. So we found out XLT4 works well for Salmonella because they can give you differential scatter signatures among different serovars of Salmonella. So initially we are interested in top 20 Salmonella serovars as published by Center for Disease Control and Prevention. We wanted to see if this light scattering technology can be used to detect any of those top 20 Salmonella serovars. As you know, the Salmonella serovars are about 2,600, and it's a high numbers to target for, so we focused only on top 20 Salmonella serovars. And when we collected the Salmonella serovars, and we had about 3 to 5 strains of each serovars, and as you can see, for a given serovar, the patterns are quite homogeneous, look very uniform. And one of the things in our earlier study, when we looked at that in St. Paul, we have 3 of those serovars produce similar scatter signatures, while there is one which is producing different types of patterns. So we were really perplexed by this result. Then we went back and looked at this Salmonella. Essentially, this is not a Salmonella sample. That was a mislabeled Salmonella serovar. And so this is also another example. In infantis, this pattern was different from the other 2 strains of Salmonella infantis. And so this indicates that this technology platform is so powerful, it can really differentiate contaminant in that Salmonella serovars. So we also compared our phenotypic light scattering pattern with the pulse field gel electrophoresis genetic pattern, and essentially the phenotypic patterns or the scatter signature patterns were similar or homologous to pulse field gel electrophoresis patterns. So they have a strong correlation between phenotype and genotype. But when we looked at also in this specific serovar, we had 2 different types of scatter signatures different from these other 2 strains. And when we looked at their pulse field gel electrophoresis patterns, indeed these 2 produced one type of PFG pattern compared to another one. So this again shows a strong relationship between phenotype and genotype. We tested if this system can be used with the real world food sample. So if the chicken sample range was taken, placed it on XLT agar plate, and you see a varieties of mixture of colonies, and some of them actually represent Salmonella, some of them are actually non-Salmonella organisms. They grow on XLT agar plate. So if you do the image library scanning, essentially you can find out the type of serovars present in the specific meat samples. We also tested that with a naturally contaminated sample. This gives you an example. These are some of the Salmonella we isolated from chicken wash sample, whereas these non-Salmonella organisms, they grow on the plate. They also can be detected on this using this system, and that data also matched well with the PFG verification we did with this study. We also tested this system with the E. coli, Shiga-toxigenic E. coli strains, and as you can see, when E. coli, Shiga-toxigenic E. coli grow on various selective agar plate, for example, rainbow or chrome agar or RNF media, they will produce different color products. And this color colony you can see only after 18 to 24 hours of growth. But when you did our light scattering, which can be done only after 12 hours, and you can see the scatter signatures for E. coli 157H7 on the plate. and they produce different types of patterns. So we used this technology and we looked at top 7 Serovars, including 157, 026, 045, and so on, and all of them actually produce highly reproducible scatter signature on sorbitol McConkey agar plate. So for different strains within the Serovar, they all produce similar patterns. So we think this technology is going to be useful for detection of different Serovars of E. coli, especially the Shiga-toxin E. coli strains. And these are the data summarizing again scatter signatures of different Serovars on different agar media. We can use this BARDOT technology for detection of E. coli 15787 directly from ground beef samples. These are the colonies that look like 15787, and from colonies from 65 through 75, these are non-15787, and you can verify these colonies by doing PCR, and as you can see, these colonies actually prove to be 15787, providing specific amplification for PCR, whereas these other colonies are non-15787. We also use this to verify the lettuce or ground beef samples when they are inoculated with 2 Serovars, 026 and 157, and as you can see, we can clearly detect these organisms from inoculated food sample, and that can also be verified or confirmed by PCR method. We also use this technology for detection of Vibrio from oyster sample, and these are some of the raw oyster, and as you can see, a lot of organisms growing on selective agar plate, and after we scan this plate, we looked for actually Vibrio vulnificus or Vibrio parahemolyticus. These are the 2 pathogenic foodborne pathogens of significant interest, and that can clearly detect these organisms in presence of background microorganisms. We also tested this for detection of Bacillus, and we were interested in Bacillus cereus group of organisms. Like before, as the Bacillus species grow on different media like tryptic soy agar or phenol red medium or modified yolk agar plate, and you can see the colony formation are different, and also their scatter signatures are also different for different species. So based on our initial study, we found out this PRM is going to be suitable for detection of Bacillus species. So essentially we tested this with the raw milk sample directly procured from market, and you place raw milk in PRM agar plate, and after probably 24-48 hours you see this type of growth, but usually within 15 hours you will see microcolonies, and after scanning with the light scattering system you can detect Bacillus cereus directly from this milk sample. So this works very well with the naturally contaminated food sample. So light scattering technology also were used for process verification purposes. In this case, our goal was to determine microbial community in, for example, iceberg lettuce obtained from US or from Mexico, and you can see the microbial profile, colony profile in those 2 different lettuce sample. And also, you could use this technology to verify if the decontamination process worked. In this case, the cantaloupe were treated by cold plasma, and so you could collect the microbial community before treatment as well as after plasma treatment. And if you look at the microbial profile, before plasma treatment, we had about 42 different types of bacterial colony diversity on this sample. After plasma treatment, you can see about 80% of them are reduced. We had only 11 different types of organisms growing. So this clearly tells you that the light scattering technology can be used to monitor process verification, and if one is interested to finding out these organisms, they can use this 16S sequencing or genome sequencing to determine the identity of these organisms. So this works very well to improve your processing conditions, so these organisms can be eliminated altogether from your products. So in summary, so as I mentioned, that biosensors can be designed for high throughput screening of harmful agents or pathogens. I talked about fiber optic sensors. This is also called immunosensors. It depends on the availability of specific antibody or DNA probe, and it cannot differentiate live from dead, however. Cell-based sensors is designed to detect only active toxin or live organisms, and it may not give you a specific information about that toxin or organism, but it can overall assess the presence or absence of harmful agents or toxins in a given samples. It's going to be also very highly powerful for detection of unknown agents, especially for bioterrorism purposes. Light scattering sensors is a label free. It doesn't require any antibody or labeling reagents. It's non-destructive, high throughput screening. It fits well with the conventional culture methods, and such as the gold standard method, and it's highly reproducible. And advantage of this method is that it will allow you to find the isolated culture which can be used for further testing. It can be used for food, air, soil, water testing. It can be used for bacterial community analysis, ecology studies, spoilage microbes. Also, it can be used for genomic library screening, and as I mentioned, effectiveness of sanitization or process verification. This also could be used in hospitals or clinics for diagnostic purposes. And it's essentially a paradigm shift instead of replacement, and we can think of using some of these biosensor technologies to help expedite the pathogen testing using existing methods, such as, like in this case, for high throughput screening. Once you have this enriched sample, placed it on the BARDOT screening for essentially finding what are the type of organisms, and you can verify each of those organisms by PCR, genome sequencing, or multi-top or spectral-based sensors. So this provides you a screening tool to rapidly assess the presence of any type of organisms or pathogens in your food test sample. Then if you are finding some of those organisms likely to be pathogens, one can use any more specific test to verify the presence of those certain organisms. I like to thank some of those funding agencies like U.S. Department of Agriculture, NSF, NIH, providing funding to do this research, as well as many collaborators. Many of them are engineers I work with to help develop these biosensor technologies. Finally, I'd like to thank Jim Lindsay. He's one of the program leaders who is instrumental in providing funding for this research, as well as my graduate students and postdocs and current graduate students, their contribution for doing many of this research. Thank you very much for your support, and I know with this I'd like to conclude my presentation. Thank you.